Hi, Dr. Pataputian. Thank you so much for taking the time to speak with me, in this like alum to alum conversation. I know you've been doing so many of these interviews, and I just really appreciate you taking the time to speak with us. So to start off, let's talk about your research. What are the big questions that you're aiming to answer? And yeah, what got you into this line of research?
So we study something that's quite ignored by most of biology, and that's mechanosensation. So the idea is your cells communicate mainly through chemicals. This is whether one cell secretes a hormone that speaks to another cell, or neurons communicate through neurotransmitters like dopamine. But what's really been ignored is how do cells sense physical force. And we knew this was very important for sense of touch, for example, sense of hearing, how you sense blood pressure, how you sense your bladder stretch. All of these we know very well, and we knew that some of it has to be very fast signaling. So this group of ion channels, they're kind of a class of proteins that are very fast signaling molecules. They sit on top of the cell, in the plasma membrane, the fatty layer that separates the outside from the inside of a cell. And they're very simple. They're either closed or doing nothing, or when they open, ions flow through. And these ions could be calcium, that could signal a variety of different things. In many neurons, sodium, for example, would depolarize the neuron and would what we call the neuron would fire. So that's the language that neurons use to speak to one another. And so it was known that there's these sensors of mechanical force that are ion channels, but their identity was not known. And so my lab found one of these proteins in 2010. We call them piezo 1 and 2, from the Greek piezi, meaning pressure. And over the last 10 years, we've been studying what they do in vivo and in real life, and both from animal models and humans. Now we know that this is how you sense touch. This is how you sense proprioception, which is that sensation of where your limbs are in space without looking at your fingertips. You can touch your nose, etc. And many other forms of mechanosensation that's important in physiology. So this includes, some of it I mentioned, how neurons sense blood pressure, bladder fullness, your lung stretch, but also some unexpected areas. And we found the role of these channels in red blood cells. And I didn't even think about this earlier, but they sense pressure as they navigate through tiny capillaries. And when they get pushed, they actually have to squeeze to go through these cells, through these arteries, those tiny capillaries. And people thought maybe this was just a passive, you get squeezed, you become smaller, but it seems like active mechanosensation is required for this process, because we know that too much piezo signaling and red blood cells are dehydrated, and very little, they're overhydrated. And then we found fascinating consequences to these, including in one case, we found that this one of these mutations in these channels that causes this dehydration also protects from malaria, which is, again, the last thing I would have thought about studying mechanosensation, but that's the beauty of basic research. You find something and science can take you in very interesting ways.
Yeah, absolutely. So just out of curiosity, the piezo channels are ion channels. So they're letting through calcium, or is it, what exactly are they letting through? And then how does that then communicate with the rest of the cell? I mean, maybe this is just too technical, but I was just curious because in reading about this, I've noticed that there are a lot of different classes of these sort of receptors. So what makes piezo unique?
Yeah. So they are what we call non-selective cation channels, which means they can let calcium, potassium, sodium through just as easily. And in different cells, it's thought different ions are doing the job. As I kind of alluded, calcium, for example, in blood vessel endothelial cells is the important signal for as a second messenger system. In neurons, it's probably a combination of that and sodium, which just changes the membrane potential of the cell, and that's enough. So, but which ions it lets through is not the unique thing about piezo. What's really unique about them is that instead of most other biology and chemistry where a chemical binds to a receptor, causes conformational change to activate, these are large proteins that spread into that fatty plasma membrane, and they're actually sensing membrane tension. So that's the really unique thing about them, is that they're the stimulus that activates them is, you know, a physical force such as pressure. Um, yeah.
Yeah, I think, are there other biological systems that respond to like that kind of physics? I've just never heard of something like that. That's not just a chemical reaction.
Yeah, so that's you know, I mentioned the hearing and touch and pain. All of these are mechanical senses, but all species experience mechanical force. We actually had a paper that a distant relative of the piezo channels in plants is expressed in plant root tips, and they actually are involved in sensing the different densities of the soil that they go through, and they help decide whether they should penetrate a surface or go around it. So it's I truly find this super remarkable that our touch sensor in our fingertips is related to something expressed in root tips to decide whether the surface is hard enough that it can penetrate or just has to skirt around it in a way.
Yeah, that's really beautiful. That kind of common shared ability within life. So, I mean, these mechanisms that you're studying are just so applicable to our daily lives. So I mean, do you like think about your own ion channels every time you hug somebody? Like, has it shaped the way that you see and perceive the world?
A little bit. At least I joke about it. Uh, um, but it actually, the reverse is also very interesting. Many scientists don't have the luxury to use intuition about their everyday experiences to drive their science forward. And that's exactly what my laboratory and I do. Like we often talk about, when I eat too big of a lunch, which I just did, I feel full, and that sounds very mechanical. You feel the swelling. And if you look at the literature of how people study satiety, being full, you know, almost all of it is by nutrition sensing, fats, proteins. These are chemicals that your body senses and decides how much you should eat, how much you should be hungry. But we know very little how that stomach stretch fullness contributes to when you stop eating. And I find that super fascinating. And again, these piezos give us kind of they're the key to enter this new area that you want to explore. And it's a molecular handle, a tool to go in and ask that question. And that's one of the directions we're actually going right now. So this is not published work, but this is something we're looking into.
Specifically the satiety, like
Yeah,
stomach.
so we're asking in animal models that don't have piezo in stomach innervating neurons, do they eat differently? Do they eat more? Do they their meal size is different? If challenged, do they become obese, or how do they respond to high fat diet in becoming diabetic, etc.?
Right. Oh, that's fascinating. Thanks. So how did you get into this line of research? Like what first piqued your interest?
So I was at Caltech. I was a graduate student working on muscle development with Barbara Wall. And when I did my postdoc, I switched to developmental neurobiology. So that was my first entry into neuroscience. And I picked the peripheral nervous system because it was less intimidating than studying the brain immediately. Peripheral nervous system being these sensory neurons that innervate your skin to tell you if it's hot, cold, or if something's touching you. And initially, as a postdoc, I was mainly studying how they develop during embryogenesis. So how does a pain neuron knows it should be a pain neuron, and a cold sensing neuron should realize it's a cold sensing neuron? How does this decided? But when I started my own lab in San Diego in 2000, just realized that in addition to that, how do they become different question. Um, this question of how do they sense temperature and pressure was even a bigger question, and the answers were not known. So decided to focus on that.
Yeah, that's great. So at Caltech, it seems like what you were working on was sort of different from what you're currently working on. So could you tell us about your time at Caltech and how that sort of shift in your career trajectory happened?
Gosh, where to start? Do you want to hear more about the science or just the experience at Caltech?
Oh, both really. I think probably more of the latter as well.
Okay. So Caltech is really where I learned to do science. And I think the most important thing you can learn in science is actually what question to ask. And what I mean by that is we all want to ask as an important question as we can tackle. But there's that, demarcation of you can be too ambitious and ask a question that today's technologies and current knowledge doesn't allow you to answer. And then there's questions that are too much detail. So I think one of the key things you learn in graduate school is what questions to ask and how to answer them. And that's what I learned the most in Barbara Wald's lab at Caltech. And so although I have changed my topic from muscle development to, how neurons sense temperature and pressure, and in science, we always are asked to change fields between graduate school and postdoc, just so that you can look at questions in very broad sense and be able to evaluate it. So it's a very important part of the process to try different areas of biology before you settle on what you want to study. So I love my years at Caltech. Used to live in this place called Prufrock House, which I still think it exists. It's this house that is surrounded by parking structures and graduate housing on Wilson Avenue, I would say. I might be forgetting the name, but so it was this place where 4 graduate students stayed. It's an old house. And we also had a cooking club where 10, 12 people would come in and we would all take turns cooking. And it was just a fantastic environment. The lab was across the street, 2 minutes walk. And I remember working very hard because I just also really loved it. So there's some very interesting guys, and Wald Lab was really fantastic environment. I have so much great memories from doing science to, running, including that there's this 24-hour relay race called the Calruf, where you team up with 10 people and run 1 mile and give the baton to your teammate. And then, but you do this for 24 hours, which means you run about once an hour for just 1 mile, which is by itself not hard, but 2 a.m. when it comes time to run again, it's pretty tough. But everything's done in a fun way. So yeah, lots of great memories. And again, most importantly, that's where I learned to do science in asking the right questions and planning to do the right experiments.
Yeah. So were you working in the Beckman Institute?
I was in Kirchhoff.
In Kirchhoff. Okay.
Yeah.
Oh yeah. So just across Wilson.
Yeah.
Gotcha. I am on Catalina right now. So I'm also just 1 block over.
Oh, okay.
Yeah.
So the
Yeah.
Catalina housing, you mean?
I don't live on campus.
Okay.
But yeah, I do live
Yeah.
on.
So Prufrock was very close to the Catalina housing. Yeah.
Right. Yeah.
It's
Yeah.
still there. Check it. It's right next to the parking structure.
Is it that strange building that kind of looks like a little medieval?
Gothic. Yeah, exactly.
I've always wondered about that
I
building.
was on that second floor. I mean, it was in bad shape. There was a huge crack on the ceiling. And I remember when the Northridge earthquake hit, I was there. So it got very, scary. But it was also a wonderful place to be.
Yeah,
and
absolutely.
has a huge history. Messelson and Stahl is this very famous molecular biologist who did some very early experiments, and I forgot which one, but one of them actually had stayed in a Prufrock house as well. I think he was in the same room as I stayed, so it was a lot of history in that building.
Yeah, oh, that's crazy. I wrote something for the magazine about the Mendelssohn Stahl experiments like a couple
Okay,
years ago, the most beautiful experiments in
that's
biology.
right. That's right.
That's so funny. So you stayed in the same house, maybe the same
Yes,
that one of them did.
yes.
Oh, wow. Caltech is such a small place sometimes.
Yeah, it is. What was
Um,
your major?
sorry.
What was your major?
I was an undergrad. I studied planetary science.
Okay.
Yeah, so I actually had no biology, well, almost no biology experience until I got this job after graduating. And the opening was for a science writer for the biology and bioengineering division. And I've just learned so much about like the living world. And honestly, if I could go back and do it again, I think I would be a biology major. Not to
Just
say that I don't love planets, but there's just so many good questions. I mean, I just love what you said about asking questions and like finding the right questions. It's just so fascinating in biology right now.
I'm
Um,
glad you share that excitement for biology.
yeah. um, okay, so let me look at my questions. Um, so thinking back on your Caltech time, is there anything that you wish you could tell your graduate student self, like any advice you would give to current graduate students, but something that maybe you could tell yourself when you were a grad student at Caltech?
Hmm. Um, I mean, I think a lot of students at Caltech at the time and now are, very stressed out about science is not easy, right? It requires hard work, but hard work is not enough. Um, you need funding, you need to publish to get to the next level. And many times I see that people let these stresses take over their lives, right? And I like to remind myself still how privileged we are to be doing research and discovery science and biology, and to come in every day to lab and asking these questions of how our bodies work at a molecular, fundamental mechanistic way. Again, I just feel like the joy of discovery is so huge. That's why we all got into it. And without minimizing the problems of the difficulties in science, it's important to constantly remind you of this, because if not, then the joy of science will go away. And if it goes away, then it's just not worth doing this job.
Mhm.
I guess that would be a
Yeah. Were there times that you felt like you lost sight of that joy or kind of got bogged down by all the
absolutely. And I think it happens more in grad school and postdoc than later as a PI, because, you know, um, we all like to think big science and think about these big questions, but on a day-to-day basis, you're sitting at the bench, pipetting very small volumes from one tube to another. And it just seems like sometimes when an experiments don't work a lot of time. So it's very easy to think this is not going anywhere, and I might be talking about doing these big experiments, but my day-to-day life is not very exciting right now. And so it does require you to stay motivated, to keep the big picture in mind while doing the experimental work in the lab. And it could be a challenge, and I certainly experienced that many times.
Yeah, definitely. So I know you've told the story of how you found out about your Nobel Prize win many times in many other interviews, but could you just tell us again about how that 2 a.m. phone call went?
Sure. So of course, I knew it was going to be announced, and I honestly thought it was going to go for the mRNA vaccines this year. But not for that reason. I always have my do not disturb on, and I wasn't going to change that. So I had do not disturb on. And I guess so Stockholm tried to call multiple times at 2 a.m., lots of missed calls,
Mhm.
and they weren't getting through to me. I don't know how they quite did this, but they somehow Googled and found another Pataputian in Southern California. And that was my father, who lives in Los Angeles, actually, is 94 years old, still lives with my mom, who's 89. And they got through to him. Interestingly, he was already awake. They called his landline and he first yelled at them, I hear, why they're bothering him at 2 a.m. But then he realized what was going on, and he called me after hanging up and said, you should pay attention to this. So in a way, it was in a roundabout way, but it's pretty cool that I heard from my dad first. And they got to me pretty much like a minute before the announcement. So usually they call you about an hour before, so you have time to get ready in a way. So I literally heard within a minute. So just grabbed the laptop and tuned in to hear the results. It was a very nice moment.
Yeah, it always seems so difficult for the people on West Coast time, just because I can't imagine that I would be coherent or even.
That was a concern. Yeah.
Did you have to take any interviews like at that time in the morning?
It started pretty quickly afterwards. The Nobel guys are pros. They know what's going to happen. They said you're going to be inundated. They said call us back in 4 hours when things have settled. And again, because you're in the East Coast already, in London, they're in the middle of the day. So I think I talked to multiple news outlets, first in London and then national press here in the United States who are in the East Coast, all before 6 a.m. my time. So yeah, lots of coffee. Well, one of the interesting things is that in the old days before COVID, when the news happened, news trucks supposedly and came to your house, and they all came to your house and did live interviews and stuff. So during COVID, at least we had that privacy. No one showed up at our place. So it was all, the press conference was on Zoom and most of the interviews are on the phone.
Gotcha. So you could still be in your pajamas from below the camera. So what made you choose to come to Caltech? I know you went to UCLA, and now you're at Scripps. And has there always been this draw to Southern California for you, or what was the kind of the deciding factor on Caltech?
So, I was before UCLA, I was an immigrant from Lebanon, some Armenian origin, but I grew up in Lebanon. And so I came to LA. It was a huge shock, of course, culturally and everything else, and had just started settling in. My parents came to LA a year and a half later. My brother was here. So one of the draws was try not to move from LA, obviously. But at the same time, I had heard of Caltech, of course, very famous place for science. And when I visited the campus, I fell in love with it. And so it was definitely my first choice when I applied. And I remember being very worried that I wasn't going to get in. So I was very, very excited when I did.
Yeah. So what drew you to the Wold Lab? Like, what about the research drew you to that lab?
I did rotations in Judy Laniel's lab and Barbara Wold's lab. I was interested in transcription, which is, and so I think what Barbara's lab was working was very, very exciting at the time. Is the idea that, again, I kind of hinted at this, how does a cell decide to become this kind of cell, right? And so that was the question that was being tackled in Barbara's lab. How does a cell decide to become a muscle cell versus a bone cell? And right when I joined, they had the field had found these transcription factors that when you put them in any naive cell, it turns it into a muscle cell.
Wow.
So there was lots of excitement about this. And I think that's where I kind of got my love for these genes that make a big impact in biology, and kind of been on the hunt of this ever since in various ways in different fields. So the field was very exciting. And I don't know if you've met Barbara, but Barbara is very impressive to talk to. She's a big thinker, and it's just a joy to talk to her, especially about science. So after my first conversation with her, I was super impressed and was looking forward to spending some time in her lab. At the same time, the lab was full of very cool, very smart graduate students who were more senior than me. Some of them I'm still good friends with. People like, Jeff Miner, Paul Garrity, and Q San Yun, all buddies that have stayed that way. And lots of times in the lab, you learn from your peers as much as you learn from the professor. So, but what I loved about Caltech the most is this really relaxed atmosphere. When you come from an undergrad school big like UCLA, you don't bump into professors all the time. They have large classes. And at Caltech, I just felt like all these very famous professors are showing up in shorts and casual wear. And every Friday we had happy hour and you could go up and talk to anyone you wanted. Again, there was this relaxed atmosphere that was really, really lovely.
Yeah, I love that too about Caltech, just feeling like you're a part of everything and not
Yeah.
like in this stratified, like
Yeah.
separate student
Yeah.
class.
Exactly. And we felt free we used to have this biology annual retreat. And I remember one year we wrote this skit and performed, like making fun of all the professors. And I still have like footage of it. It's on old VHS tape or something, but it's just, you could see all the professors laughing out loud at themselves. It was just great environment.
Oh, that's so wholesome. So how has your life changed since you got the Nobel Prize, or has it changed at all other than all of these Zoom interviews?
It has. I'm trying for it not to change too much, and I'm honestly just processing all this yet. It's I think over the next few years I'll have a more clear answer of how I want this to affect it. So in one hand, I don't want it to affect it because I love my life before. I love the discovery. I love being in the lab, and I don't want this to change too much. But at the same time, this does give you opportunity to actually give back. One of the things I mentioned already, that I'm Armenian origin, came from Lebanon, and before this, there was no Nobel Prize to either someone with Armenian origin or from Lebanon. And so they've really embraced this as their own, which I find it lovely. And so, for me not to give back, or when people say, if you come to Armenia and talk to the youth and the scientists, they'll be inspired that they can, you can't really say no to that. And so I think one or two causes, I'm a huge advocate of immigrants in the US and how they are treated. One interesting example is, actually I haven't shared this before, but I did 1 year of American University of Beirut and then came to UCLA. For a number of years in my CV, I had not, you don't have to write this, I'm just sharing it with you. I hadn't written the AUB part because I was concerned that the Beirut, Lebanon would put people off, that, oh, he's from a terrorist country, right? So I kind of didn't put that, and I thought legally it was okay because I never got a degree from there, so I justified it, right? Now after this, I go out of my way to describe it because I just feel like now I don't feel like I have to hide it anymore, or that I want to bring some positive news to those issues and countries, that you can be from that background and still succeed. So it's kind of changed my perspective a little bit on it. And again, whether it's immigrants in science or giving back to the Armenian community or whatever, there are a few things I'm looking forward to being part of that's outside the lab.
Yeah, that's great. Thank you for sharing that. That's really beautiful. Well, I think that was all of the questions that I had. Was there anything else, regarding your Caltech experience or how your time at Caltech may have shaped your career that you wanted to share?
Um, no, I mean, one of the big things for me at Caltech was also that's where I met my wife. So she was an undergrad at Caltech, and she was a senior when I was starting in grad school, and so she also worked in the World Lab a little bit, and we've been together since. So Caltech has special meaning for me for that reason as well. So she went to Berkeley for grad school while I was finishing up. So for 3 years we were apart, but then we met up in Bay Area, and now, yeah, down here in San Diego. So Caltech is not only has given me started my career, but also personal life. Yeah.
Oh, that's great. Well, thanks so much again, Artem. And it was really great to meet you. And I just, ever since I wrote this press release about your work, it's just really made me think about the way I interact with the world, and I touch things and sense things. And anytime I use, the peppermint shampoo, I'm like, oh, sensations of cold.
That's really wonderful for me to hear. Thanks for thanks for
Yeah,
telling me that.
of course.